This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, episode number 235, recorded on January 28th, 2021. I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today from Small Things Considered, Elio Schaechter.
Well, hello there. How are you?
I'm very well, and you?
I live in a cold state. Southern California turned cold on me. Can you imagine?
Oh, toughen up, Elio.
It'll go back.
Michele, tell him the temp in Michigan.
Well, we had single digits last night, but it's in the 20s today. But a lovely snow cover.
Lovely.
Yeah.
It's a winter wonderland.
Also joining us from Ann Arbor, Michigan, Michele Swanson.
Hello, everyone.
A little bit of snow, like a dusting you have there?
No, I'd say maybe an inch and a half.
Very good. And from Charleston, South Carolina, Michael Schmidt.
Hello, everyone.
Is it balmy there?
Nah, it's about 50s.
Oh, tough.
It's
very
about
tough.
50s.
That is balmy.
That
It's
is
tough.
balmy. And the sun came out. It's been raining the last 2 days, and the sun has finally come out and brightened everything up. So I turned off the lights on the desk, and you can see the room is pretty bright.
Michael, you have a tie on today. That's unusual for home, right?
That's because I did a story for one of our local reporters over double masking. That was today's topic. Double masking, as well as I sat for an interview with the Washington Post, and that story is running today.
Very good. Good to talk with knowledgeable people, right?
We
Yes.
try.
So, should we double mask?
Yeah. What did you say to that, Michael? Tell us.
I said that
What's the punchline?
the punchline is an additional mask will reduce the risk.
Okay. Got
How
it.
much? How much? 1%? I mean, what is it?
I didn't give him a percentage, and I said because it's filtration. If you put it through a second filter, it will filter more.
I figure since people don't even wear their first mask properly, maybe that'll help, right?
That's the issue. I said you have to be wearing it properly. Everything leaks, I said.
Everything gets out of control.
Let me say that I've started to put one of the blue kind of surgical disposables on underneath my cloth decorative. Yeah, one of those.
Yeah.
And I actually find it more comfortable to have that first layer. It's a bit absorbent.
And then you throw it out. You toss it at the end of the day. Yeah. Okay. That's what I'll do.
And it fits in well. You know, I cut the loops. I cut the strings off, so I'm not dealing with two layers of loops around my head. So it's not a big deal.
All right. So we have two stories for you today, and I'm going to do a snippet. And Elio this week sent out two papers, and one of them was about viruses. So guess which one I chose?
The virus one.
You know, I mean,
we're lucky to have you.
I have done non-virus papers here on TWiM, but this was quite interesting. It's a Nature article entitled "Widespread Endogenization of Giant Viruses Shapes Genomes of Green Algae." And Elio, were you taken to this or drawn to this because of the algae?
Not particularly. I was drawn to it because it's a crazy story. In a sense, it's a crazy story.
I see. Okay. So this comes out of...
I thought it fits you.
It fits me because it's crazy. Yeah. Thank you very much. Yeah. It comes out of Virginia Tech, which is in Virginia. I guess you would guess that, right? And the authors are Moniruzaman, Weinheimer, Martinez Gutierrez, and Alward. And this is very interesting because everyone knows that, or everyone should know that when retroviruses infect cells, their RNA is made into DNA by a viral enzyme, which then integrates into the nuclear DNA of the host. And if the host, if that cell doesn't die, the DNA can persist. And if that cell is a germ cell, then we say the retroviral DNA becomes endogenized.
It's about 7%. Isn't it about 7% of the total?
Yeah, our genome is 7% retroviral. That's right, from ancient
big deal.
Ancient infections, and then during evolution, our bodies have taken over some of those viral proteins. It's a process called exaption, EXAPTION. We have exapted the viral gene. So, for example, one of the genes needed to make a placenta, it codes a protein called syncytin, is a former retroviral spike glycoprotein. It's very cool. And there are other examples as well. And not
Spike
just
comes back,
not just proteins, but promoter elements, regulatory elements. All of these have been exapted. So that's retroviruses. Okay. Now it turns out that since we're sequencing so many genomes of hosts, you can ask, are there viral genomes integrated? And there are. They're not complete genomes. So the difference here is, and this is key, for the retroviruses, integration of DNA into the host is obligatory for their reproduction. Virus can't reproduce without inserting its DNA into the host cell. But for all these other insertions, endogenous viral elements, they're called, or Eves, right? Eve. We'll get to Adam at some point, but this is Eve. The insertions are random bits, and the virus doesn't require them. They just happen accidentally. But if they go into a germline, then they will be passed on. And we can see in many genomes, including ours, pieces of all kinds of viruses, both DNA and RNA viruses. Okay. Again, the key is those are not needed for virus reproduction. Only the retroviruses are. So this paper is about green algae, which can be infected by giant viruses. And, in the viral world, this has been an amazing discovery that there are really, really big viruses with genomes over 1 million base pairs of double-stranded DNA. They infect amoeba, green algae, and many other organisms. And so what they did in this paper is ask, are there any integrations of these big viruses into various organisms like green algae? And they do find them. And it's mainly a computational biology exercise where they write a program and say, okay, here are sequences of giant virus genomes. Find them in these cellular genomes.
I wonder what made him do it. What is supposed to make him do it?
Well, because no one else had done it. And the data are all public. You can find all the sequence data out there. And it's waiting to be done. Frankly, as you would say, seek and ye shall find, right? And they saw it.
True.
It's not a bad project to take on when the labs are at low capacity or sometimes closed.
Yeah.
Right. You can do it from the comfort of your home.
That's right.
And you can assign students to learn computational work, and it can be pretty fascinating. This reminds me of the novel by Greg Bear called Darwin's Radio. Did any of you ever read that book? It's about endogenous retroviruses changing the human race into a super race. It's science fiction. It's a novel.
Let's hope.
But
Let's
it's got the retroviral elements in our DNA, and magic happens, and we have developed superpowers.
Well, I have to say,
maybe link to it in the show notes, Michael.
I'll do it.
Retroviruses have made us able to have a placenta, and
That's
without
true.
that, we'd be laying eggs.
Some of us, not all of us.
Yeah, about half of the population.
Well, we all had to have a placenta at one point in time in order to get born, right?
Yeah, but we don't actually make one. Guys don't make them.
Our mothers did.
But without that, I like to say, we'd be laying eggs. How about that?
True.
So that's transformed humanity.
Chlorella.
No, anyway, Elio, to answer your question, they say our knowledge of the prevalence of Eves that originate from double-stranded DNA viruses remains relatively unexplored. People have done this mostly for RNA viruses. And so that's what drove them. So these giant viruses are called NCLDVs, which they put in the introduction, but they don't define it. How can you do that? How can you put a term like that? Anyway, nucleocytoplasmic large DNA containing viruses. That's what that stands for. And that's where all these giant viruses are lumped into, because the name is very important. Nucleocytoplasmic. Some of them go right into the nucleus, but some of them stay in the cytoplasm, and some of them do both nucleus and cytoplasm. All right, so they do computational biology. They look in chlorophyta, which is a group of green algae related to land plants. And there are many important viruses. There are many big viruses that infect members of chlorophyta, including Chlorella. I don't know if any of you know Jim. What's the guy's name in Nebraska now? I'm forgetting his name, and I have to look it up because he's just a cool guy. Jim Van Etten. James Van Etten.
Yes.
You must know him, Elio, right?
No, I don't. I've run into his work, though.
All right. So he's been studying virus of Chlorella for ages, and that was actually one of the first big viruses after the pox viruses that were discovered. He's a mountain of information about them. Really wonderful. Mm. So what they did here is they took 65 genomes of these host cells, 6 classes within the chlorophyta. And in 24 of these, they identified genome signatures of these giant viruses. And again, they have very big genomes. And these big pieces, so basically these are pieces of the viral DNA that have been integrated into the genomes of these chlorophyta. And they ranged in size from 78 base pairs, very short, to 1,900 kb. All right. So they could tell in many cases what viruses they were. They found them in 12.
That's bigger than some free living organisms.
Yes. So that's the
and
cool
big enough
thing
to
here.
encode a protein.
So many of these are big enough to encode a protein. And in fact, they contain between 76 and 1,700 predicted genes. And so, one of the key observations here is that these cells are getting a lot of extra DNA from this,
Man, are they ever.
So there's one chlorophyta, T. socialis. There are 2, they call them GEVs now, giant endogenous viral elements, GEVs. That used to be a butler, right? Jeeves, I think.
Or a search engine.
That's right. It was a search engine. So they say there's 2 Jeeves in T. socialis that have 2,800 genes, and they represent 10% of the cell genome comes from the virus integration, right? Now, not only do they find them there, but then they see duplications of these viral genes and gene loss. So in other words, these happened a while ago, and then there have been genome rearrangements occurring over time. And many of these Jeeves are missing genes, and probably none of them are functional, I would think. They didn't look because this is just a computer study, but they mentioned later that many essential genes are often missing. So it's not likely that these DNAs integrated into the cell are making viruses at all.
So this whole thing may be for nothing.
Well, that's a good question, right? So is it for nothing or not? Now, in other cells, people are asking this question. And one possibility is that, okay, first of all, if you maintain a gene for millions of years, is that because it doesn't matter or because it means something, right? So some people think, well, if you make some viral proteins, maybe it will defend the cell against virus infection. There's some evidence for that in mammalian samples that we've looked at here. But they basically say we don't think these are making viruses, these Jeeves, but we just think the genes are present. And that's what these EVEs are in general. They're pieces of genomes because integrating is not essential. But the question is, what is it? As Elio says, is it meaningless, or is there enough room in a genome that you can have extra stuff and it doesn't matter? Or do these mean something? So someone at some point needs to do the experiment. And this is a kind of observation that might, trigger someone to do something.
Well, in a larger host, it likely doesn't impose a fitness penalty on it because it has sufficient energy in order to replicate the DNA. But in a virus, it would likely impose a fitness penalty on it to carry all this excess baggage. And it just needs what the eukaryotic cells need is a good editor to effectively get rid of the extraneous information that it's no longer making use of or doesn't add to the story.
Yeah.
But there's potential, depending on where it integrates, it could disrupt a function or a regulatory pathway, or it could change a regulatory pathway or a trait and confer a fitness advantage and contribute to evolution and maybe speciation.
Absolutely,
So,
yes.
But it's probably all of the above is happening because this is a random event, right?
It is a random
And I
event,
actually like Michele's argument that it confers a fitness advantage to carry on this extra information because remember, evolution is a stochastic process that is effectively seeing what will allow the host to get its genes to the next generation. And, it's all stochastic.
So the last thing I want to tell you is the last paragraph, they say, well, bacteriophage integration into bacterial genomes is known to be a driver of genomic innovations, right? These viruses can bring in genes that do something for the host. They can transfer them among hosts. And they say it's been thought that this is less common in eukaryotes, but they say, hey, maybe it's not so common. And, there's some evidence that these giant viruses actually transfer genes among hosts. And so, it's probably wrong to think it only happens in bacteria. Yeah. I mean, bacteria may have invented it, but why would a eukaryote throw it out, right? Hey, look at this gift I have, virus-mediated gene transfer. Why would I get rid of it? So
it's a very provocative observation.
They conclude by saying this kind of endogenization represents an underappreciated aspect of eukaryotic genome evolution and suggests that many eukaryotic lineages have access to a much larger array of genomic material than previously thought. So, cool. I mean, if you can add 10% more of your genome with a virus, that's amazing, right? So, that's
we got to stay tuned in because a lot of the things are going to show up.
Oh, yeah.
And I guess it's a nice thing about bioinformatics. It's cheap.
It is cheap. It is cheap. You just have to pay the salary of the people doing it, right? Which usually is the biggest cost of any research project.
That's true.
Right. But it does stimulate further experiments, right? Then people say, oh, let's do an experiment and see if this gene is transcribed, if the protein is made. You could do that. So much to do. So little time, so few people. I'll end it on that. Money. All right.
Yes.
Now for our paper, we turn it over to Michael.
Yes, this is a manuscript that comes to us from the University of Texas Southwestern Medical Center in Dallas. It's by Ellerman, Jimenez, Pfeiffer, Ruiz, and Sperandio. And Ellerman and Jimenez equally contributed to this work, and the first author order was determined as Melissa Ellerman actually helped supervise some of the early experiments of Angel Jimenez. So this is a very interesting, and it appeared in MBio this month. So it's in the public domain. So you have access to it via the ASM's online journal portal. This is a very interesting story about microbes, specifically bacteria, how they appreciate what's going on in their environment. If you will, this paper is going to be a bread and butter regulatory paper where we investigate, and this is the title of the paper, the canonical long-chain fatty acid sensing machine, where the canonical long-chain fatty acid sensing machinery processes arachidonic acid to inhibit virulence in enterohemorrhagic E. coli. And so this machine that is able to sense this long-chain fatty acid, arachidonic acid, you need not be a microbiologist to appreciate that the mammalian gastrointestinal tract, where enterohemorrhagic E. coli acts, is a complex machine. But what the general public and non-microbiologists may not appreciate is that bacterial pathogens are continuously sensing and responding to specific stimuli within their gut that are in turn used to coordinate the regulation of their virulence programming. I have been teaching these past few weeks about virulence and pathogenesis in infectious disease course for dental students. So I've been thinking a lot about how to instill why understanding the mechanics of virulence programming is important for the next generation of clinicians. Not necessarily to teach them the science, but rather teach them what it means. Teach them what it means clinically. And, often you get asked the questions, why should we care? Or is this going to be on the test? But in reality, it really is very important to understand how stuff works so we can figure workarounds. So let's consider this title again. Canonical long-chain fatty acid sensing machinery processes arachidonic acid to inhibit virulence in this bacterium, enterohemorrhagic E. coli. Now, EHEC, which is the abbreviation, is different from the cloning strain of E. coli, for it has acquired a toxin gene, and it's only appropriate that Vincent talked about bringing in genes from our neighbors. Well, this gram-negative, where it acquired the toxin gene, was from Shigella, another Enterobacteriaceae, but in a completely different genus. And Shigella is only naturally found in gorillas and humans, but in humans, it causes an estimated 80 to 165 million cases of shigellosis each year, with the number of deaths tragically being estimated between 75 and 600,000.
Which is a painful, bloody diarrhea.
Oh, yes, yes. So what is arachidonic acid? Our colleagues over on TWiM, and forgive me, Vincent, but you guys have been doing this. The colleagues over on TWiM have as one of their many story arcs this meme that when they speak of chemicals such as arachidonic acid, they often refer to them as 5-hydroxy chicken wire because they look like chicken wire. That's what arachidonic acid looks like, chicken wire. If you've ever seen a chicken coop, you've seen arachidonic acid. So they do that on TWiM to convey what the chemistry looks like. Well, arachidonic acid fits into this club. It's a polyunsaturated omega-6 fatty acid. It's got 20 carbons. So it's big. It's got lots of energy, and it's structurally related to the saturated arachidic acid found in capucho butter, which is not from milk, but rather from the cold pressing of seeds from species that's native to Brazil, Theobroma grandiflorium. And according to this is why I'm digressing, but according to the internet, it's the secret to healthy, hydrated, and moisturized hair. And I can use all the help to hold on to the hair I currently have. So arachidonic acid, being 20 carbons, is uniquely present in mammalian, but not bacterial membranes. Now, in response to environmental stimuli, arachidonic acid is liberated from the membrane of the host and the membrane-associated phospholipids and monoacylglycerols, and it acts as an important host signaling molecule that modulates a diverse range of host functions. If you will, it's one of the nuclear weapons of the host in combating microbes in our gut. Now, from the perspective of the microbe, arachidonic acid also exerts this potent growth inhibitory effects on numerous gram-positives and gram-negative pathogens through mechanisms that involve oxidative stress and disruption of membrane integrity. So back to why this is a relevant question. So with all of this as prelude, Ellerman and Jimenez and their colleagues demonstrated that the family of exogenous, this means in the lumen of the gut, long-chain fatty acids, can directly inhibit the locus of enterocyte effacement, or the Lee regulatory element on the pathogenicity island in this pathogen of E. coli through a sensor molecule or a sensor circuit called FADR. Now, one final bit of background before we get to the meat of the paper is that this group had previously identified the transcription factor, FADR. So as you are familiar, transcription factors typically interact with a nucleic acid in order to affect a behavior. They either turn on something or they turn off. And the transcription factor is activated when you have these long chain fatty acids that have been activated by coenzyme A. And so the coenzyme A interacts with it, and that then determines the virulence factor's regulator. It effectively activates it. And this is unique to the enterohemorrhagic E. coli. Now, polyunsaturated fatty acids play, again, a critical role in host immunity. And in fact, we have witnessed the manipulation of our lipid content within our tissues through diet. You know, everyone has heard thou shalt not eat bacon, or the pharmacological interventions, take your Lipitor. And that's associated with the severity of various inflammatory diseases. So as we move through the data, I want you to keep at a high level that the work here illustrates a defined host pathogen interaction where arachidonic acid, a host-derived and a dietary polyunsaturated fatty acid, can impact the infection of this enteric pathogen, or EHEC. Up front, there is more microbial biochemistry in this paper than we have talked about in a while. So I struggled with how much to include without doing a disservice to the authors, but trying to spare your brains as you're listening along. But then again, I'm nerdly about biochemistry because it helps me to understand how stuff works. So,
and Michael, if I could interject, the authors
Sure.
Wisely included a very nice schematic as panel A of figure 3 that lays out this regulatory pathway that includes the FAD regulators that you'll be talking about. So for those of you following along at home, you might want to open up to figure 3A.
And it really opens up the paper for you if you just follow along. Now, again, trying to think about how do you explain the type of disease that this is causing at a local level inside the gut of the host. Well, we know about type 3 secretion systems being present in the gram-negative, and they confer typically virulence functions. The type 3 secretion apparatus we've talked about on TWiM before, and we know it makes a needle-like device that injects bacterial proteins into our cells that actually causes behavior. And so as the name of this molecular circuit implies, the locus of enterocyte effacement, that's effectively what happens to the host, is their enterocytes are effectively being effaced by poor E. coli. So EHEC utilizes this Lee-encoded type 3 secretion system to first promote physical attachment to the epithelium by modifying the epithelial cytoskeleton and injecting its cognate receptor, TIR, to intimately attach to the epithelial membrane and form a pedestal-like structure. Now, Brett Finley did a tremendous video animation that's in the public domain for the 1999 holiday lecture series, 2000 and beyond, confronting the microbial menace, that I'm going to put into the show notes so you can follow the animation that really puts it into perspective of what this bacterium is actually doing in the gut and how arachidonic acid is interfering with this process of effectively putting E. coli on a throne. So what happens?
And the wonderful animation from the Finley lab includes an amazing soundtrack as well. So I highly recommend, if you do nothing else, watch this video. It's amazing.
Now you have to appreciate that Dr. Finley is Canadian, so he uses words like wiener-like structure to refer to microvilli. And they really do look like hot dogs. Canadians call hot dogs wieners, and we call them hot dogs in the U.S. So what you see is that the proteins that E. coli has put into our enterocyte effectively create this through the action of actin, this pedestal that effectively raises E. coli up on a throne into the lumen, which makes it more available for nutrients, which results in this characteristic attaching and effacing lesion that then results in this painful and bloody diarrhea that Michele introduced us to in the beginning. So we all appreciate that there are a variety of signals in the gut from the host, whether they be sugars, whether they be peptides, whether they be lipids that are sensed by EHEC. And they are integrated into a complex signaling cascade that regulates this Lee pathogenicity island. I'm going to refer to this locus of enterocyte effacement from now on as Lee, simply because it's one abbreviation instead of 3 words. So in the first figure, the authors walk us through the transcriptional activation of Lee. So first up is LAIR, the master regulator, which is the first gene contained within Lee1. We learn from the authors as background that this regulatory region is heavily trafficked by different transcription factors that each have the ability to sense and respond to specific stimuli. So again, the stimuli are interacting with transcription factors, changing their ability to hold on to the nucleic acid, which in turn activates or represses the transcription of the operon. So we have the transcriptional regulation of LAIR-R and the entire Lee island. As I said, this has been previously published, and the authors give you good guides to their references so you can go back and check out. And I bring this to your attention so you can appreciate the next bits. Now,
and also another really nice color-coded schematic to introduce this pathogenicity island.
Well, I think that's one of the reasons that I was attracted to this paper. It was so well laid out, and their diagrams are so informative for both the skilled and naive to folks who look at transcriptional maps and trying to understand what's going on. So a long-chain fatty acid is non-esterified, and operationally it's defined, it contains 12 or more carbons. This becomes relevant because arachidonic acid is 20 carbons. One of the other long-chain fatty acids they look at is palmitate, which is 16 carbons in length. So E. coli will bring in long-chain fatty acids across its outer membrane through, again, a transport mechanism facilitated by a protein called FAD-L. And this effectively grabs the long-chain fatty acid, it gets it across the hydrophilic periplasm, and then it flips across the inner membrane to its inner leaflet. Once inside, these long-chain fatty acids get activated by coenzyme A, which then allows the cell to chew on long-chain fatty acids like a piece of beef jerky. They nibble 2 residues at a time through a process of beta oxidation. Remember, beta oxidation whacks off 2 carbons just as if you took a bite out of the beef jerky, and your bites were always perfect in 2 carbons in length, which in turn coordinates the consumption as well as the synthesis of fatty acids that E. coli needs to make more E. coli. Now, one of the things that I failed to mention in our prelude, about arachidonic acid is that there are several Enterobacteriaceae species, such as coli and others, but there's also Pseudomonas, and they exhibit resistance against the bactericidal effects of arachidonic acid. And in one of their figures, figure 2 to be precise, they actually show how arachidonic acid can actually inhibit the growth of Staph aureus. And it's a black and white figure that you just add the AA to the culture, growing it under the right conditions, and boom, it stops the growth. So again, we go back to the title of the paper, namely, arachidonic acid inhibits virulence of enterohemorrhagic E. coli. We're not talking about growth. We're talking about virulence. So this is where we begin with the authors of the asking the question, what does exposure of arachidonic acid do to the expression of this Lee pathogenicity island? So again, look at figure 3, look at figure 1, and you'll appreciate that this Lee island in EHEC is controlled by Lair with the first bit of this multi-gene locus pathogenicity island. They had previously reported that other long-chain fatty acids, the long-chain fatty acid CoA responsive transcription factor, FAD-R, directly binds DNA targets located upstream from the Lee1 promoter. So they next asked whether if you add long-chain fatty acids, will it impact EHEC virulence by transcriptionally modulating Lee? So the first thing they try is 16 carbons or palmitate or palmitic acid. And this interacts with FAD-R when activated by the acyl-CoA synthetase, which is effectively going to tack on CoA to the fatty acid that comes in. And that's effectively this they measure at micromolar concentrations of palmitate with decreased expression of Lee as measured by the expression of a gene product called ESPA, which, if you don't have the map, is all the way at the end of the circuit. It's at the rightmost end of the molecular circuit diagram that they provide. And this encodes a secreted protein that forms a filament that wraps around the type 3 secretion needle. Again, go back to the animation from Brett Finley, and it will help you understand what these magic proteins like ESPA and the other ESPs are actually doing. Now, the conclusion that they reach from this first set of experiments is the long-chain fatty acid palmitate inhibits the pathogenicity island in EHEC. So next up—
at the level of transcription.
At the level of transcription. So next up was the question of whether the host's nuclear weapon, arachidonic acid, a polyunsaturated fatty acid that is present and it's liberated from the mammalian gut in response to an enteric infection, will have a similar effect. So when the host-derived arachidonic acid was present, the transcription of the representative genes from each of the 5 Lee operons was significantly decreased. And this is expressed in one of the first figure 1D. And they offer us a figure where they compare vehicle alone, because in order to get the long-chain fatty acids in, they have to have a vehicle that will get them into solution. And the vehicle alone, and they really do beautifully controlled experiments. Bottom lining this, though, from a clinical perspective, is that with reduced functionality of a type 3 secretion system, as measured by the production of ESPA and ESPB, and you can judge for yourself in figure 1E, you can imagine that virulence will be reduced as E. coli is not able to express the full virulence profile of this leukocyte, enterocyte effacing program. So this observation that they have made in the first figure was observed over a range of physiological concentrations of arachidonic acid. But here's the twist in answer to the questions why we recall hamburger that has the enteric with the Shiga toxin in it, in spite of the fact that our host system has arachidonic acid to inhibit this Lee pathogenicity island, is that the arachidonic acid that our host is making in response to this infection does not appear to exert an overall antivirulence effect on EHEC, the whole organism in total, not just the little Lee pathogenicity island, because transcription of the phage-encoded virulence factor, Shiga toxin, which causes the hemolytic uremic syndrome, was not altered. And they snuck that into their supplementary section of their data. So you'll have to go look for that cool experiment there. Now back to
so that observation allowed them to zero in then on the interaction of these long-chain fatty acids with the Lee pathogenicity island specifically. Right?
Yes, it winnows things down. And maybe, you know, in the pre-show, Elio was asking, how do you explain canonical? You know, why is it canonical? Well, I offered that it was this regulatory element is regulated by many signals in E. coli. And so that could be why they use the term canonical. And they really want to demonstrate that arachidonic acid inhibits virulence in this particular situation. So their next experiment established that inhibition of the locus, or Lee, by arachidonic acid is not dependent on growth because, as I told you previously, arachidonic acid will turn Staph aureus completely off. And remember, Staph aureus is another gut pathogen. Any of you who have ever had staphylococcal food poisoning know how well that causes projectile vomiting. So, well, it does.
I believe you.
I was once sitting in an airport listening to a flight attendant and a pilot talk about their recent food poisoning experience at one of the airport restaurants. And they described projectile vomiting. And we then had a discussion about staph because they looked at me and they told me it happens. And they told me not to eat there. So looking back to this really cool science, arachidonic acid is this potent antibacterial compound against numerous bacteria at micromolar concentrations. So the host doesn't have to dump out much. So here the experiment they offer us is whether this antivirulence effects of arachidonic acid in EHEC occur through bacteriostatic or bactericidal mechanism. So they cultivated EHEC under microaerobic and aerobic conditions in the presence of physiological concentrations of arachidonic acid. And what they found is arachidonic acid did not impact EHEC growth. And they beautifully show that in the first panel of the second figure, where EHEC at various concentrations of arachidonic acid, they mirror the vehicle alone growth curves. And it's really quite remarkable just looking at the various concentrations they offer us. And similarly, it doesn't appear to impact the growth kinetics in any way whatsoever. And then the third panel of that figure, or 2C, they show that it doesn't impact on the viability of it. So we're not controlling virulence by killing the enterohemorrhagic E. coli. We're doing something else. So that's piece of evidence number one. So they conclude from the experiments associated with the second figure that the inhibitory effects of arachidonic acid on this regulatory system are unlikely to be due to any detrimental or promotional effects on the growth of this organism. Next, they offered us how arachidonic acid is influencing this locus of enterocyte effacement. So again, they remind us that they're sensed by FAD-R, a transcription regulator subsequent its transport of the arachidonic acid into the cell by FAD-L. And again, this now takes us to the third figure where they give us first in panel A their beautiful color-coded map of how long-chain fatty acids are sensed by E. coli in this particular case. And they describe how the repression of Lee is dependent upon FAD-R. Now, the problem with these complex bread and butter regulatory players is that you need a glossary with you, unless you're in the field, of who does what to keep things straight. But the authors do a fine job of trying to keep us on the straight and narrow, referring us back to previous work and really expanding on what's actually going on here. So again, they start with a little bit of background about Enterobacteriaceae fluctuations in the intracellular concentrations of these long-chain fatty acids are assessed by the FAD-R transcriptional regulator. So many of you are familiar with cyclic AMP serving to activate the CAP protein in the lac operon, where it becomes a transcriptional activator if and only if there's enough cyclic AMP around. So for the sake of argument, think of the long-chain fatty acid as being cyclic AMP. It's not, but just if you understand the lac operon, you'll sort of understand where we're going with this. So when the cyclic AMP tickles CAP, it then becomes a better positive regulator of transcription in E. coli. And that's similarly what's happening here. But in this case, instead of it being called CAP, it's called FAD-R. Now, all of this occurs subsequent to the exogenous long-chain fatty acid being sent across the membrane through the transport of FAD-L, which you see in figure 3A, which is, again, the molecular circuit diagram. So naturally, their next experiment is to tinker with the transport system to tease apart whether components of the long-chain fatty acid sensing pathway are required for arachidonic acid to exhibit its inhibitory effects on Lee. So step one, construct a mutant of FAD-L and assess the effects on arachidonic acid on Lee-dependent type 3 secretion activity. So they do the mutations, they lay out the beautiful experiments, and they showed us that arachidonic acid failed to repress Lee in the FAD-R mutant. So in order to rule out the possibility that beta oxidation contributed to the anti-virulence effects, if you will, the eating of the beef jerky of the arachidonic acid, they tested the effects of arachidonic acid on Lee-dependent type 3 secretion activity in an isogenic FAD-E mutant, which is deficient in the first enzyme that induces beta oxidation. So you can't eat the arachidonic acid. Here again, as with the parental strain, the FAD-E mutant also exhibited decreased ESPA and ESBB secretion in response to both palmitic acid and arachidonic acid. Now, importantly, no growth effects were observed in FAD-L or FAD-R or FAD-E isogenic mutants under control conditions. So this represents a lot of work. They offer us additional data in their supplemental figures. And rolling this up, they conclude that these mutant studies, from these mutant studies, that both FAD-L, the transporter, and FAD-R, the regulator, are required for arachidonic acid, the 20 carbon fatty acid, and palmitate, the 16 carbon fatty acid, to exert there are anti-virulence effects on this Lee circuit in enterohemorrhagic E. coli through a mechanism that is not dependent upon the consumption of the fatty acid. So in the next experiment, they begin to put things.
Wait. I think we're getting into a little bit too much detail now.
All right, then I'll jump to the final experiment in support of their title. So this is where arachidonic acid. So let me set up the experiment
briefly.
The hallmark of EHEC infection is the formation of these attaching and effacing lesions, making of the pedestals, if you will. You can see these if you do a colonoscopy. You can actually look at them, and they actually show us some beautiful confocal microscope shots where they light up. They have the appropriate probes, and they can see it. To demonstrate that arachidonic acid can inhibit EHEC infection and pedestal formation, they first pre-treat EHEC with arachidonic acid or the vehicle control. And then they infect epithelial cells, and they ask a question, do you form a pedestal? They look by confocal microscopy, which is figure 7A. And it just is sitting there waving at you. It's this bright color. They observed that pedestal formation was reduced by approximately 50% with arachidonic acid treated EHEC. And they also assessed it by the infection rate. And the data are just exquisite. Not only do you see the vehicle, which has this effacing lesion with these little dots that you can see on their tissue culture images, but then you add arachidonic acid and poof, all the dots are gone. And they just lay it out so it's so crystal clear.
It's
Summing
really
up,
lovely.
all of their findings support a model where arachidonic acid is recognized by this circuit, Lee. It's processed and sensed in machinery in this enterohemorrhagic E. coli. This ultimately diminishes DNA binding by fat R, thus attenuating the ability to transcriptionally activate the Lee operon. And remember, the Lee operon, the Lee1 operon, is what starts making the syringe and the lethal injection proteins that cause the pathology that is ultimately responsible for the bloody diarrhea. And finally, they offer that their findings demonstrate in that in addition to knowing its growth inhibitory effects reported for things like Staph aureus, arachidonic acid also acts as a signal that can modulate the expression of canonical genes involved in long-chain fatty acid metabolism and catabolism that in turn exerts an anti-virulence effect in enterohemorrhagic E. coli. There's a lot more data in this paper, but as Elio said, it's taxing to your ears to follow along without making notes. And many of you listen to us as you're running or driving. And so I apologize to the authors for shortchanging them, but I commend you to go take a look at this paper. It's fantastic.
So should we take arachidonic acid when we're at risk of having EHEC?
Yes.
I think, like everything else with any antimicrobial, moderation is the key, because you could disrupt other things. Like there are important gram positives in your gut, and we don't know what is going on with them with respect to arachidonic acid. And for the most part, our food supply is relatively free of enterohemorrhagic E. coli. And it's only in those bad times when you get a bad lot of food that it affects some folks. But we do understand how to. Go ahead,
Do you think
Michele.
this pathway only benefits the host by blocking that stage of the pathogenesis, or might might EHEC be advantaged? Because presumably they would want to not stick there on this pedestal forever. To release and then move on to another part of the intestine might be an advantage. So if the initial microcolony makes the pedestal and causes enough tissue damage locally that then the arachidonic acid levels build up and then act as
Shuts
a negative feedback. Yeah, and now the microcolony could lift off, would lift off perhaps, or stop causing damage, maybe spread to another spot.
Or diarrhea. The peristaltic flow of diarrhea actually moves it to other parts of the colon. So you're expanding the niche, or it just moves out to another new host through the process.
But they move more readily if they weren't making pedestals, right?
Correct, because the pedestal allows them to attach.
Yeah, so I'm wondering if this brings up the age-old saying, the goal of every microbe is to become two microbes, not necessarily to cause nasty disease. So I'm wondering if this is an example of kind of the cat and mouse between the pathogen and the host, and there is some advantage to EHEC to have this sensor of tissue damage?
I think that gets back to the important bit in their title. This long chain fatty acid sensing molecular circuit interprets the host defense to the initial infection insult, and it actually shuts it down. But it all goes back to a discussion about fitness. What allows the bacterium to become a second bacterium? And I think that ultimately is what drives much of virulence, much of pathogenesis, and much of the host continuous tour de force between host and microbe.
Luckily, there was a really talented team working on this beautiful project and interesting biology. And so if I could tell you a bit about Melissa Ellerman, the first author. She was a postdoc when she did this work with Vanessa Sperandino's lab. And her story is that as a high schooler, she was interested in interior design, graphic design. So when she started at Rochester Institute of Technology, she thought that's what she would major in. But then she became interested in green design. She's took some classes in environmental science. And then in her junior year, she took a really interesting biology class and as part of the biotechnology degree program, and found that it was just fascinating because of the passionate and engaging teachers that she had in that biology class. So her favorite classes in the end at RIT were microbiology and virology. She loves the diversity of living organisms and the interactions between human microbes and our immune system and that coevolution. So she then went on and got a PhD in microbiology and immunology at UNC Chapel Hill. And in her first rotation, she was introduced for the first time to the gut microbiota. So that just blew her mind. She ended up joining Balfour Sartor's lab and worked in a mouse model studying the effects of diet on the gut microbiota and how that affects Crohn's disease. So she continued that work in her postdoc with Vanessa Sperandino, and just 3 weeks ago, she started her dream job. She's an assistant professor in the Department of Biological Sciences at the University of South Carolina. And she's currently teaching an undergraduate microbiology class, which she recognizes is the very class that inspired her own research trajectory. So I'm sure that's motivating her to share her passion about microbiology with the undergraduates and the next generation. So her advice to younger scientists is to be open-minded. Don't be afraid to explore your interests. Let them guide you, and don't feel like you're going off track if your career path or your training takes you on a winding path, because that's how you're going to find out what you're really excited about and what inspires you. And I'll close by saying that Melissa has been a TWiM listener for some time, and she was really excited to hear that we'd be featuring her paper today.
Nice.
And she mentored the second author, who Elio was going to describe?
I'll tell you a little bit about the second author. Both authors have an equal role to play here, so we're doing both. His name is Ángel Jiménez, and Ángel was born in what he calls the beautiful island of Puerto Rico, and he got into microbiology not because he got a chemistry set when he was young, but he just had a light for nature. And this was emphasized by going on a trip in the bioluminescence bay of an island of Puerto Rico called Vieques. If you go there at night and you dip your oar in the water, it lights up. And he thought this was amazing. These are dinoflagellates doing that. Anyhow, he got into microbiology. To make it brief, he worked with Melissa Edelman, and they did said she was an incredible mentor. Anyhow, he's advised to young scientists. By the way, he's involved in minority issues in microbiology, in science, which is wonderful. So he says, I encourage aspiring scientists to keep their values on top of mind and never waver in your character. Keep curiosity as a driving force in your scientific endeavors, and be brave and fearless in your scientific approach. There is no experiment that is beyond your abilities. There's always a solution to the questions at hand. Golden
Wow.
words.
Nice. Brave and fearless. I like that. It's very
Brave,
good.
fearless.
Not sure I'm
And
brave
he's
and
currently
fearless.
now a postdoc at UT Southwestern. And according to his LinkedIn page, he aspires to a career at the intersection of science and policy.
Ooh.
Yeah. Nice.
Good luck with that. Good luck. All right. Thank you, Elio. Thank you, Michele. And two quick emails to wrap this up. One is from David, who sends us a joke.
A joke.
Have you ever heard
Why
the
not?
one? Have you ever heard the one about the salmonella outbreak? It was death by 1,000 cold cuts.
Oh,
That would work for listeria too, wouldn't it?
Got you guys to laugh. That's good. And then one from Martin, who is from the University of Namibia in Africa. I'm a first-year medicinal chemistry student, and I would like to pursue microbiology next year because I have more interest in being able to find out about a certain disease and its cure, or just explore the beauty of microbiology like you guys do on your podcast. Any advice you have for me? Because in my country, it seems like science, it's not really supported the most. Therefore, we don't have things like this here, but I would like to learn how to establish an association. So I'm not sure what you want to do, Martin. It's not clear if you want to do science or you just want to talk about it. You know, if science isn't supported in your country and you want to do science, you probably have to go somewhere else, which may not be easy. However, if you just want to talk about microbiology, go on social media, go on Facebook, go on Twitter, and talk to people about it. Find some groups that talk about microbiology and do that. I think that's a thing you can do now that we couldn't do, years ago.
Another option is to explore if there are local chapters of, for example, the American Society of Microbiology. They have ambassadors throughout the world, and that might be a way to spend more time with people doing work that interests you and get some ideas about how you might get involved.
That's a good idea. All right, that'll do it for TWiM 235. You can find the show notes at microbe.tv/TWiM. If you want to send us a question or a comment or even a joke, that's good. It works.
Hey, you can use jokes.
TWiM at microbe.tv. And if you like what we do, consider supporting us financially. We have a number of supporters. We appreciate their donations. Microbe.tv/contribute. Michele Swanson's at the University of Michigan. Thank you, Michele.
Thank you all. Great to see you again.
Elio Schaechter's at Small Things Considered. Thank you, Elio.
My pleasure. Thank you.
Michael Schmidt's at the Medical University of South Carolina. Thank you, Michael.
Thanks, everyone.
I'm Vincent Racaniello. You can find me at virology.blog. I'd like to thank the American Society for Microbiology for their support of TWiM and Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.